So in the previous video, we were able to knock out the first 3 learning goals on your screen. In this video, we are going to talk about 3 more learning goals. The first one being describing homeostasis and learning a little bit about how the endocrine system uses feedback loops. We'll talk about negative feedback and we'll talk about positive feedback and how the endocrine system uses these loops to control hormone release and keep the body in balance. We're also going to describe hormone functions. We're going to look at classifications by chemical composition, and we'll talk about the mechanisms of action on target cells. Finally, we will describe the formation, release, and physiological effects of thyroid hormones. Homeostasis describes the ability to maintain the internal environment within parameters that are designed to maintain and sustain life. And typically, we're going to define those parameters as conditions that fall within normal limits. And so, for example, blood pressure. If the blood pressure increases, the body is going to secrete certain hormones to try and decrease the blood pressure. There may be some other structural things that happen. There may be dilation or expansion of our blood vessels, or constriction and reduction in diameter of our blood vessels in response to some of the physiological changes that our body may experience. And so why is this important? If homeostasis is not maintained, then our cells cannot function in the way that they were designed to function. And so, for example, if the pH of our blood is too high or too low, it could affect the way our enzymes function. It could cause our proteins to break down and therefore they will not function. Extreme temperatures could also denature proteins and prevent their expression. If we have a situation like hypoxia or lack of oxygen, this could lead to tissue damage and organ failure. High or low blood sugar could also affect energy levels. And so homeostasis is very important. Everything that I just mentioned could turn fatal if not resolved in a reasonable amount of time. The endocrine system primarily uses negative feedback loops to regulate hormone levels and maintain homeostasis. And so basically what happens is a change that occurs within the body that is not normal, the body is going to try and reverse that change. It's going to try to do something that kind of move things into the opposite direction, okay? And so, for example, if we have blood glucose levels that rise, then the body may release insulin to try and lower blood glucose levels, okay? When we look at body temperature, when our body temperature declines, then we may shiver as a way to generate some heat and try to bring us back up to the normal range. And if our body temperature goes above that of what is normal, the body may sweat to try and cool down and release moisture to help get it back to the normal range, okay? Here on your screen, get my laser here, this is a nice little illustration that kind of shows you step by step an endocrine process that occurs with regards to blood glucose levels. When we're eating, we're going to expect a spike in our blood glucose levels because we're consuming something that has glucose or that's going to be broken down into glucose. And that is going to cause the pancreas to release insulin, as you can see here. Insulin is going to signal to the cells, take in the glucose, because our blood glucose levels are too high and we need to lower them. And they will. And when they do, we have reduced blood glucose levels, as you can see here. And the presence of normal blood glucose levels are going to turn off all of these factors here that caused us to take in more glucose into our cells, okay? So by having a normal blood glucose level, we're not going to have insulin being released by our pancreas, okay? So basically everything that's happening here would be the opposite, okay, once we have our normal levels, all right? And then on this side, we have fasting. If we are in a situation where our blood glucose levels fall too low, it could signal the pancreas to release glucagon and reduce the amount of insulin that's being released, okay? And so insulin is going to target different parts of the body. One place that it's going to target is going to be the liver, where we can release glucagon, or not glucagon, but glycogen that can be broken down into glucose and be deposited into the blood to raise our blood glucose levels, okay? And so that's what you see here. Now, positive feedback loops are not as popular as negative feedback loops in regulation within the endocrine system, but there are some scenarios where we might see it, okay? And basically with positive feedback, we just have a response that is amplified, okay? And so if we have a stimulus that generates a response, the way that the brain is going to respond is it's going to send out more hormones that are just going to amplify that response until the result of what we're trying to achieve is accomplished, okay? And so the action of the effector is the thing that causes the change, that intensifies the stimulus. This is going to lead to a deviation from a change, okay? This is going to cause a reinforcement or amplification of the changes. So rather than oppose the change, the response is going to be in the same direction as the change. So the example on your screen, we have a baby within the womb of a mother that is pressing against the cervix, and this in itself is an effector. This is causing a signal to be transmitted to the brain about something going on. And so the brain is going to respond by releasing oxytocin. Oxytocin is going to travel by way of the bloodstream to the uterus, and it's going to promote and stimulate uterine contractions and push the baby through and towards the cervix, okay? This is a situation where we don't want negative feedback. We don't want to get to a point where we stop contractions because we've met a threshold, okay? The goal is to successfully deliver the baby, and so positive feedback, we are going to continue and continue and continue to pump out oxytocin to try and get those uterine contractions, try to get them stronger so that we end up with a delivered baby, okay? We also see this with voltage-gated sodium channels. In order for sodium channels to remain open, the membrane potential has to be at different set points. I'll use that for lack of better words. And so when we have like an influx of sodium, or if we have a situation where we have an efflux of potassium, that can affect the opening and closing of those channels, okay? With lactation, the baby suckling on the nipple of its mom could send a signal to the brain letting it know, hey, we got to feed a baby here. We need milk. And so that's going to result in the release of prolactin from the brain to target the breast tissue, so that, or the ducts within the breast tissue, so that more milk can be produced to nourish the baby. And so those are just some examples of where positive feedback is used. 2 examples specifically on your screen where it is used in the endocrine system. Hormones are going to exert their effects by altering metabolic processes. And hormones that are activated may result in cellular responses such as opening or closing ion channels, stimulating enzyme or protein synthesis, activating or deactivating enzymes. It can induce secretory activity. It can stimulate mitosis. So there's a lot of different activities, and it's not just limited to what you see on your screen. These are just examples of how the hormones can alter metabolic processes within the body. Now, there are 2 major mechanisms by which hormones bring about their effects on their target cells through interacting with the receptor on the plasma membrane if the hormone is water soluble, or entering the target cell and activating the receptors inside if the hormone is lipid soluble, okay? And we're going to focus on the first way in the next couple of slides. So we're going to be looking at water-soluble hormones, which are going to be all amino acid-based hormones except thyroid hormones. They are going to act on the plasma membrane receptors, so they do not enter into the cell. If you think about the plasma membrane of the cell as a phospholipid bilayer, so they're not going to have an easy time getting in the cell anyway. So the receptor must be present on the membrane surface. They're going to act via G protein second messengers. Okay, and G proteins, just a side note, are called guanosine binding proteins. Okay, that's just your fun fact of the day, in case you wanted to know. All right. So non-steroid hormones, amino acid-based hormones, with the exception of thyroid hormone, cannot enter into the cell. Okay, they have to act on the surface of the cell. As mentioned on the previous slide, water-soluble hormones are going to exert their effects through second messenger systems. And there are two main second messenger systems we're going to talk about in this class. We're going to go over the processes involved with the cyclic AMP system, and then we're going to talk about the PIP2 calcium system. All right, so let's look at cyclic AMP and their signaling mechanism. And I'm talking to you and you see a whole bunch of words, so if it's a lot, just give me a moment, bear with me. Hear it first, and then I'm going to show you a video, or not a video, but an illustration of it later, okay? So we have our first messenger, the hormone, and it's going to bind to a G protein coupled receptor located on the plasma membrane, okay? The receptor is going to change its shape, and when it does that, it has the ability to bind to an inactive G protein, okay? And there are different G proteins that are located very close to the plasma membrane within the cytosol. There are G proteins that can activate other enzymes, and then there are G proteins that can inhibit other enzymes, okay? And so the enzyme in question is going to be our effector enzyme, adenylate cyclase, okay? So the G protein that's activated, that has the ability to activate adenylate cyclase, is going to lead to the formation of our second messenger, okay? So adenylate cyclase is going to catalyze the formation of CAMP, which is our second messenger, from ATP. It converts ATP into CAMP, okay? Now CAMP is going to activate protein kinases that are going to have the ability to phosphorylate other proteins. And why is that important? Proteins that are phosphorylated by those protein kinases can either be activated or inactivated, okay? And so the protein kinases allows us to be able to lead to either the expression and functions of those proteins or not, all right? To maintain homeostasis, phosphodiesterase is an enzyme that can rapidly degrade CAMP, stopping the cascade stopping the cascade of the protein kinases from phosphorylating other proteins. Okay. CAMP activated hormones include amino acid based hormones like epinephrine, antidiuretic hormone, adrenocorticotropic hormone, luteinizing hormone, follicular stimulating hormone, glucagon, some of our autocrines like prostaglandins. And so these are just some examples of hormones that use the CAMP system to activate their proteins. All right, so let's follow our running men, or our key players up top here, and we're going to follow them together. I'm get my laser here. We have our hormone, which is our first messenger. We have our receptor, okay? We have our G protein. We have our enzyme. And do you guys remember what our second messenger was? CAMP, okay? So if you want to put a little note here, right, that is CAMP, all right? So in this first frame, we have the hormone right here is going to bind to the G protein coupled receptor, okay? This receptor is going to change shape and it's going to open and expose its site so that our GTP bound G protein can bind to the receptor, okay? Then the receptor is going to activate that G protein, okay? That G protein is then going to bind to adenylate cyclase. And adenylate cyclase is going to use ATP to create CAMP, all right? And then CAMP is going to activate protein kinases that are going to phosphorylate proteins and allow them to be expressed, okay? So this is going to trigger a series of responses at the target cell and to include, but are not limited to, activating the enzymes, stimulating cellular secretion, opening ion channels, etc. Okay. Not all water-soluble hormones will use the CAMP signaling mechanism. Some water-soluble hormones may use the PIP2 calcium signaling mechanism, where when the hormone binds to the receptor on the plasma membrane and activates that G protein, instead of activating adenylate cyclase, it activates a different enzyme, phospholipase C. Okay, and phospholipase C has the ability to split the membrane protein PIP2 into 2 second messengers. Okay, so the 2 messengers that it splits into is diacylglycerol, which is DAG, and that's going to activate protein kinases that are going to be able to phosphorylate other proteins within the cell. Okay, the second messenger, or the second messenger that it splits into is inositol triphosphate, IP3, and this is going to trigger the release of calcium from the endoplasmic reticulum within the cell. Calcium ions can also act as another second messenger. Calcium has the ability to affect the activity of some proteins, but more times than not, it's going to do so with some help, okay? And so calcium binds to a protein called calmodulin, okay, creating a complex consisting of calmodulin and 4 calcium ions. This configuration allows it to bind to protein kinases and activate them, okay? And so this is going to cause phosphorylation of those proteins and their activation or inhibition depending on kind of what the function is of the particular protein kinases that are activated. An example of a PIP activated hormone would be oxytocin. We'll spend some time talking about lipid-soluble hormones, which are going to be steroid and thyroid hormones. They have the ability, due to their chemical composition, to be able to enter into the cells. And so that's what they do. They enter into the cells, they act on receptors within the cells, and then they can activate genes. And when those genes are transcribed and translated, they can create proteins that can go and do what needs to be done, depending on the function of the hormone. Okay, so we'll kind of go through this step by step and what this looks like. Okay, so we have the steroid hormone here that enters through the plasma membrane, doesn't have an issue doing so, and it finds a receptor and it binds to the receptor, forming a receptor hormone complex. Okay, the hormone complex is going to enter through and into the nucleus. And it's going to look for a segment of DNA. Okay, its target DNA is target gene, and these words are all on top of each other. I apologize for that, but it's just saying that the receptor bound hormone is binding to a specific region on the DNA. Okay, binding is going to initiate transcription of the gene. And so basically a copy of this DNA is made into RNA. We call that mRNA, okay, during transcription, all right? The mRNA transcript is going to leave out of the nucleus into the cytoplasm, where we're going to have a process called translation. And during this process, we just create a new protein, Okay, and so once that protein is created, it can be activated so that it can carry out the functions of the cell. Okay. All right, so let's look at how thyroid hormone is synthesized. While we're talking about thyroid hormone and how it enters into the cell because it's a lipid-soluble hormone, um, just to kind of introduce you, this is a histological sample, a tissue sample of the thyroid. We have this lumen space here that is filled up with the fluid called colloid, and this is where parts of what will be thyroid hormone goes to get processed. And then the precursors to thyroid hormone is contained within these cells surrounding the colloid, the fluid. And these cells are called follicular cells, okay? And again, they contain the precursors to thyroid hormone, all right? When we look over here, we have the follicular cell, okay? Or sorry, back up. We have the colloid. So where I am kind of drawing an area with my laser, this is colloid, okay? And then we have the colloid right here, okay? And then we have the bloodstream here. This side is the apical surface of the cell. And over here, we have the basolateral surface of the cell, okay? And so I'm going to go through the steps of how we make thyroid hormone, and I'm going to try not to be super technical about this and kind of how this works. You may hear some of the terms listed on your screen. I may or may not use them. It just depends on kind of how deep or simple I go in terms of explaining this. But I just wanted it to be there in case you hear something and you're like, I don't know how to spell that, but I want to know what it is. I want to get it right in my notes. It is here. If it is not here, then it should be down here somewhere. Okay. All right. So the very first part of making thyroid hormone is synthesizing a chemical called thyroglobulin. Okay. And the follicular cells of the thyroid can synthesize thyroglobulin, and then it's thyroglobulin that is produced by the follicular cells is going to be processed by the endoplasmic reticulum here before it is packaged up by the Golgi to go into the colloid. Okay, so once it's packaged up into the vesicle, it's going to leave by way of exocytosis, as you can see here, and go into the colloid where it is going to be further processed. Okay. In the second step, we have iodide trapping. And so iodide is going to be actively transported by an active transport pump. This pump, we call it the sodium iodide symporter. Okay. And so iodide is going to be pumped from the bloodstream by way of the symporter into the cell. Okay. For every iodide ion that enters into the cell, we are able to pump in 2 sodium ions. Okay. The next thing that happens is iodide is going to be oxidized to iodine, okay, by the enzyme thyroid peroxidase. So you see that here, okay. Once it is oxidized, is also going to enter into the colloid space, okay? Because eventually we're going to add these iodine molecules to our newly created thyroglobulin molecule here, okay? In the next step, iodine is going to bind to the tyrosine residues of the thyroglobulin molecule. And so when it does that, it's going to form 2 different molecules. It's going to form MIT, monoiodotyrosine, and it's going to form DIT, diiodotyrosine. Okay? In the next step, we have coupling. And so the MIT and DIT molecules can be bound together to form thyroid hormones. There's 2 thyroid hormones that can be formed. We can form T3, which is MIT and DIT bound together. We call that triiodothyronine. the second thyroid hormone that we can create is called T4, and it is created by binding DIT to another DIT molecule. And that is going to create T4, which is also called thyroxine. Okay. Now, once we have created the reaction where we are binding iodine to thyroglobulin containing T3 and T4, we are ready to take it back into the cell. Okay. So by way of endocytosis, as you can see that here, we are going to take in that processed thyroglobulin, the T3 and T4. Excuse me. And once it gets in there, it's going to be broken down by lysosomes, and it's going to allow for T3 and T4 to be released into the bloodstream. Okay. Now, most T3 and T4 are going to bind to plasma proteins for transport to target tissues. Okay. Great. So you've made it to the end. I know thyroid hormone synthesis was a little, it was a lot. There was a lot of steps, and I hope that I did a good job in helping you understand it. But if not, please ask questions. I do have a question on here about thyroid hormone, and I have a couple others about other topics that we talked about in this video. But please take a moment to kind of review the questions, review your notes, and please use me as a resource if you have anything that wasn't clear or you need clarification on.